It is August 1st, 2024. A baby boy is born at the Children's Hospital of Philadelphia. His parents, Kyle and Nicole Muldoon, are expecting a healthy fourth child, a little brother for their three older children. The birth goes well. The baby is small, just 4 pounds, but he is here and he is theirs, and his name is Kyle Jr. KJ. Within hours, the nurses notice something wrong. The baby is unusually sleepy. He is not feeding properly. There is a lethargy about him that does not belong to a newborn, something beyond ordinary tiredness, something that feels, to the experienced eyes of the neonatal unit, like a warning. A blood test is ordered. The results come back and a doctor looks at them and has to look again. KJ's ammonia level is over 1,000 micromoles per liter. Normal is between 9 and 33. KJ's level is more than 30 times the upper limit of normal. Ammonia, at these concentrations, crosses the blood-brain barrier and begins to destroy neural tissue. At this level, in a newborn, the damage can begin within hours. KJ is transferred to the neonatal intensive care unit. Genetic testing begins. The results take days, and in those days his parents wait in the way that parents wait when their child is connected to monitors and surrounded by the quiet urgency of a medical team that is working very hard and saying very carefully chosen words. The diagnosis arrives: CPS1 deficiency, carbamyl phosphate synthetase 1 deficiency, a genetic disorder so rare that it affects approximately 1 in every 1.3 million people worldwide. A disorder in which the liver cannot process ammonia, the toxic byproduct of the protein metabolism that every human body performs every day. Without treatment, the ammonia builds up and the brain pays the price. There is a treatment, a liver transplant, but KJ is a newborn. The transplant cannot be done until he is at least a year old, and in the year it would take to reach transplant eligibility, the ammonia would keep accumulating and the damage would keep compounding, and what arrived on the other side of that year might not be the same child who entered it. His parents sat with this information. And then a doctor walked into the room and said something that no doctor had ever said to any parent in the history of medicine. She said, "There is another option. We can rewrite his DNA." This is the story of KJ Muldoon, the first human being in history to be treated with a personalized, custom-designed gene-editing therapy built specifically for him, targeting the exact mutation in his unique genome. The story of the scientists who built it in 6 months, when the estimate was 18. The story of the parents who said yes to something that had never been done. And the story of what it means for every child who comes after him. This is the story of the day medicine changed forever. To understand what KJ Muldoon faced, and what the treatment that saved him actually did, we need to spend some time understanding the biology. Every cell in the human body is constantly breaking down proteins. It is one of the most fundamental processes of metabolism. Proteins are consumed, digested, and their constituent amino acids are either recycled or further broken down. When amino acids are broken down, one of the byproducts is ammonia, a compound that, at low concentrations, the body handles easily, but that at high concentrations is profoundly toxic, particularly to the brain. The body's primary mechanism for disposing of ammonia is the urea cycle, a series of chemical reactions that take place primarily in the liver, converting ammonia into urea, which is then safely excreted in the urine. The urea cycle is one of the most important metabolic pathways in the human body. Without it, every protein you eat, every piece of chicken, every glass of milk, every egg produces a toxic byproduct that your body cannot remove. The first and most critical enzyme in the urea cycle is called carbamyl phosphate synthetase 1, CPS1. It is, in a sense, the gatekeeper of the process. Without CPS1, the urea cycle cannot begin, and ammonia cannot be converted to urea. KJ Muldoon was born without functional CPS1. The genetic testing revealed why. He had inherited two different mutations in the gene that codes for CPS1, one from his father, one from his mother. The mutation from his father was called Q335X. The mutation from his mother was called E714X. Each mutation alone might not have been catastrophic. People can sometimes function with one normal copy of the CPS1 gene and one mutated copy. But KJ had no normal copies. Both of his CPS1 genes were broken. His liver could not begin the urea cycle. It could not convert ammonia to urea. Every protein he consumed produced ammonia that had nowhere to go. The standard treatment for severe CPS1 deficiency is a liver transplant, replacing the damaged liver with one that has functional CPS1 genes and can perform the urea cycle normally. But liver transplants are major surgery, requiring significant physical size and development in the recipient, and they are generally not performed on infants younger than approximately 1 year. They also require lifelong immunosuppression, drugs that prevent the body from rejecting the new organ, which carry their own serious long-term risks. In the meantime, CPS1 patients are managed with an extremely restricted protein diet because protein consumption produces ammonia. Reducing protein reduces ammonia production. They are also given medications called nitrogen scavengers, drugs that help the body dispose of ammonia through alternative pathways. These are imperfect solutions. The protein restriction is severe enough to risk affecting normal growth and development. The medications have side effects, and neither addresses the underlying problem, the broken gene. For KJ, who had one of the most severe forms of the disease, even these management strategies were barely sufficient. His ammonia levels remained dangerously volatile. His diet was so restricted that normal infant development was at risk. He spent the first months of his life in the hospital, connected to monitors, on a diet that no infant should have to sustain, waiting for a body that was too small for the surgery that might help him. And then Dr. Rebecca Arens-Nicholas walked into his life. Dr. Rebecca Arens-Nicholas is a physician-scientist at the Children's Hospital of Philadelphia, a doctor who both treats patients and conducts research, bridging the gap between the laboratory and the bedside that is often wider than it should be in modern medicine. She had been thinking about CPS1 deficiency and gene editing for years before KJ was born. She had watched children with urea cycle disorders suffer through the limitations of existing treatments, the restricted diets, the metabolic crises, the uncertain outcomes of liver transplantation in infancy. And she had been watching the development of a technology that she believed might one day change what was possible for these children. That technology was CRISPR. CRISPR, clustered regularly interspaced short palindromic repeats, is a gene editing system derived from a natural defense mechanism found in bacteria. In bacteria, CRISPR sequences are used to recognize and cut the DNA of invading viruses, disabling them. Scientists realized in the early 2010s that this system could be adapted into a precise molecular tool for editing DNA in any organism, including humans. The key insight was this. The CRISPR system uses a small piece of RNA, called a guide RNA, that is programmed with a specific genetic sequence, like a GPS coordinate in the genome. The guide RNA directs a cutting enzyme, called Cas9, or in more recent versions, a base editor, to the exact location in the DNA where the edit needs to be made. Once there, the enzyme makes the targeted change. In 2020, Jennifer Doudna and Emmanuel Charpentier received the Nobel Prize in Chemistry for their development of CRISPR gene editing. It was one of the most consequential scientific achievements of the 21st century. The The technology had already been used in clinical trials to treat certain blood disorders, sickle cell disease, beta thalassemia, by editing cells outside the body and returning them to the patient. But what Ahrens Nicholas was thinking about was different, more ambitious, more dangerous, and more powerful. She was thinking about editing genes inside the body, in vivo, as scientists call it, delivering the gene editing machinery directly to the liver cells where CPS1 is supposed to be expressed, and rewriting the broken genetic code right there in place, without removing the cells from the body. This approach had never been done in a human being. There were significant technical and safety challenges. The gene editing machinery needed to be delivered to the liver, specifically to the liver, without going elsewhere in the body. It needed to make the correct edit and not introduce errors elsewhere in the genome. And it needed to be safe, in an infant whose immune system and metabolism were already under severe stress. Ahrens Nicholas had been working on this problem in the laboratory since 2023, in collaboration with Dr. Curran Musunuru, a cardiologist and geneticist at the University of Pennsylvania's Perelman School of Medicine, who had been developing CRISPR-based treatments for liver-related conditions, including high cholesterol. They were not working toward a specific patient. They were working toward a platform, a system that could, in principle, be adapted to treat any patient with any mutation in a liver-expressed gene. And then KJ Muldoon was born. When KJ's genetic mutations were identified in the days after his birth, Ahrens Nicholas recognized immediately what she was looking at: CPS1 deficiency, two specific mutations, a severe neonatal onset case with a very limited window of opportunity. She called Musunuru. What followed was one of the most extraordinary collaborative sprints in the history of medicine. The challenge was specific and enormous. Design a gene-editing therapy targeting KJ's specific mutations, the Q335X mutation from his father and the E714X mutation from his mother. Manufacture it in sufficient quantities and to sufficient purity standards. Test it for safety and efficacy in laboratory conditions. Obtain regulatory approval from the FDA, and administer it to a child who might not have months to wait. The estimated timeline from the Vice President of Integrated DNA Technologies, one of the companies involved in the manufacturing, was 18 months. They did it in 6. The approach they chose was not the original CRISPR-Cas9 system, which cuts both strands of the DNA double helix, a powerful but somewhat blunt instrument that can introduce errors. Instead, they used a more recent and more precise technology called base editing. Base editing is, in a sense, a more surgical version of CRISPR. Rather than cutting the DNA and relying on the cell's repair machinery to fix it, a process that can introduce errors, base editing uses a modified enzyme to directly convert one DNA letter into another. The 4 letters of the genetic code, A, T, C, and G, can, with base editing, be swapped one for another with extraordinary precision, at a specific location in the genome, without cutting the double helix. For KJ's Q335X mutation, the specific base edit required was the conversion of a single letter, changing a C to a T at the precise position in the CPS1 gene where the mutation had occurred. This single letter change at this precise position would restore the genetic code to something the cell could read correctly, allowing the production of functional CPS1 enzyme. The therapy was designed as messenger RNA, genetic instructions, packaged in a fatty bubble called a lipid nanoparticle, that could be injected into the bloodstream and delivered specifically to liver cells. This delivery system was not new. Lipid nanoparticles had been used successfully in the COVID-19 mRNA vaccines, giving them an important safety track record. But the cargo was unprecedented. A custom-designed base editor paired with a custom-designed guide RNA, targeting a mutation that existed in only one person on Earth. The therapy was given a name, KJ Muldoon, or more simply, K-Abe after KJ himself. The regulatory pathway was equally unprecedented. Nicholas and Musunuru applied to the FDA for a single-patient expanded access investigational new drug designation, a compassionate use pathway designed for critically ill patients with no other options. It is, as one medical journalist described it, not formal research. It is designed to help a patient. The FDA issued approval in 1 week. 1 week. From application to approval for a never-before-tested gene-editing therapy in a human infant. This extraordinary speed reflects both the severity of KJ's condition and the quality and thoroughness of the application that Nicholas and Musunuru submitted, backed by years of preclinical research, rigorous safety testing, and the collaborative input of institutions including the innovative Genomics Institute at UC Berkeley, the Broad Institute of MIT and Harvard, and Massachusetts General Hospital. The institutions involved worked without financial compensation for their contributions. The pharmaceutical companies provided in-kind support. The scientists worked nights and weekends. Sandy Ottensman, the vice president at Integrated DNA Technologies who led the manufacturing effort, later said simply, "We estimated it would take 18 months. We did it in 6. On February 25, 2025, 6 months after KJ Muldoon was born, the first dose of K-Abe was infused into his bloodstream. He was 6 months old. He was the first human being in history to receive a personalized gene-editing therapy. The day of KJ's first infusion was, by all accounts, one of the most tense and most extraordinary days in the history of the Children's Hospital of Philadelphia. The therapy had been designed for KJ. It had been tested in the laboratory. It had been approved by the FDA. Every precaution that science and medicine could devise had been taken. And still, nobody knew, in the absolute sense, what was going to happen when this unprecedented treatment entered this specific child's body for the first time. The infusion was scheduled to take 3 hours. Nicholas and Musunuru were in the room. Kyle and Nicole Muldoon were in the room. The monitoring equipment was running. The nurses were ready. The therapy, packaged in its lipid nanoparticles, carrying its custom-designed molecular machinery, was prepared. KJ slept, while his parents and his doctors sat with the weight of everything that was happening, in the particular silence of a hospital room where something unprecedented is occurring. The baby at the center of it all simply slept, peacefully, unbothered. Those 3 hours, by the account of everyone present, were among the longest of their lives. The infusion completed without incident. KJ showed no immediate adverse reaction. His vital signs remained stable. His ammonia levels did not spike. The lipid nanoparticles, carrying their base-editing cargo, had entered his bloodstream and begun their journey to his liver cells. The real question, whether the base editor was finding its target, making its edit, and restoring some degree of CPS1 function, would take weeks to answer. The liver cells that received the edit would need time to begin producing functional CPS1 enzyme. The change, if it was working, would gradually manifest as a change in KJ's metabolic markers, his ammonia tolerance, his ability to process protein, his need for nitrogen scavenger medications. KJ received a second dose in March 2025 and a third in April, the maximum number of doses permitted by the FDA under the compassionate use designation. And then they waited. Nicole Muldoon, speaking later about this period, described it with the specific eloquence of someone who had lived through 10 months of fight-or-flight intensity and was beginning, carefully, to believe that something different might be possible. She said, "We've been operating in fight or flight for so long. It's starting to be like the light at the end of the tunnel." Kyle described his own journey through those months with a detail that has stayed with everyone who heard it. When KJ was first diagnosed, he had gone home briefly and found a Philadelphia Eagles jersey he had ordered before the birth for his son, who he suddenly wasn't sure would ever wear it. He said, "I forgot I even ordered it. I just sat here and I cried and I was like, I just don't know if he's ever gonna wear this." The results, when they came, were better than the team had dared to hope. Within weeks of the first infusion, KJ's metabolic markers began to change. His ammonia levels became more stable. He was able to tolerate more protein in his diet, a significant indicator, because protein tolerance is the direct measure of CPS1 function. More protein can be consumed only if more ammonia is being processed, and more ammonia can only be processed if more CPS1 enzyme is being produced. His need for nitrogen scavenger medications, the drugs that had been helping his body manage the ammonia backup, began to decrease. This was another direct indicator. Less medication needed means less backup occurring, means more functional CPS1 activity. He began to reach developmental milestones. He sat upright. He grew. The 4-pound peanut of August 2024 became, by the spring of 2025, what his mother described as a big, chunky, thriving baby, a transformation that was not just physical but symbolic of everything the treatment had made possible. As of April 2025, KJ had received all 3 doses of KABE with no serious adverse effects. On May 15, 2025, Dr. Curran Musunuru stood before a packed auditorium at the American Society for Gene and Cell Therapy annual meeting and presented KJ's case to the scientific world. It was published simultaneously in the New England Journal of Medicine, one of the most prestigious medical journals in the world. The scientific community's response was immediate and extraordinary. Musunuru told the room and the world that he did not think he was exaggerating when he said that this is the future of medicine. He was not exaggerating. On June 2, 2025, 307 days after he had first been admitted to the Children's Hospital of Philadelphia, on the day after he turned 10 months old, KJ Muldoon went home. He had spent his entire life to that point in the hospital. He had never been home. He had never slept in the room his parents had prepared for him, surrounded by the ordinary objects of an ordinary baby's life. The hospital gave him a send-off that reflected what the staff felt about this child and what he represented. The hallway outside his room was lined with nurses, doctors, technicians, and support staff who had cared for him over those 10 months. There were bubbles. There were familiar faces. Nicole Muldoon said later that when she walked out of his room and saw the entire hallway just lined with people, she couldn't help but cry. He was escorted home by a police escort. He wore his Eagles jersey. The story of KJ Muldoon is, on one level, a beautiful human story, a baby, a family, a team of dedicated scientists, a treatment that worked. But it is also, at a deeper level, a story about a scientific revolution that has been building for decades, and that is now, with KJ's case, beginning to deliver on its most ambitious promises. Let us spend some time with the science, because understanding it is essential to understanding why what happened to KJ matters so much, and what it means for the future. The human genome contains approximately 3 billion base pairs, 3 billion letters of the genetic code, organized into roughly 20,000 genes. Each gene contains the instructions for making a specific protein. Mutations, errors in the genetic code, can disrupt the instructions, producing proteins that do not work correctly or no protein at all. There are approximately 7,000 known rare genetic diseases, conditions caused by specific mutations in specific genes. Most of them have no cure. Many have no effective treatment at all. They are the medical frontier, the conditions that pharmaceutical development, which is driven by market economics, has largely passed over because the patient populations are too small to justify the investment in treatment development. CPS1 deficiency affects roughly 1 in 1.3 million people. The entire global population of CPS1 patients at any given time is probably in the hundreds. No pharmaceutical company will develop a treatment for hundreds of patients. The economics do not work. And this is where the personalized medicine revolution that KJ's case represents becomes truly radical. CRISPR, and specifically base editing, is what scientists call a programmable technology. The guide RNA that directs the editing machinery to the correct location in the genome can be designed for any sequence. It is like a GPS that can be programmed with any destination. You identify the mutation. You design the guide RNA to match the surrounding sequence. You build the base editor to make the specific change needed. And you have a therapy. The specific guide RNA that was designed for KJ will not work for any other patient, because no other patient has exactly his mutations, in exactly his genomic context. KABE is, in the most literal possible sense, a therapy for one person. But the platform, the base editor, the lipid nanoparticle delivery system, the manufacturing process, the regulatory pathway is reusable. The next patient with a liver metabolic disorder will not need 18 months or 6 months. The platform exists. The regulatory relationship exists. The manufacturing capabilities exist. The knowledge exists. Dr. Musunuru, reflecting on this after KJ's case was made public, said something that captures the full scope of what has been accomplished. The goal, ultimately, is to partner with the FDA to establish a robust gene editing platform that can be deployed to treat any inborn error of metabolism in the liver, so that nobody will need to undertake a heroic effort to help a child like KJ. It will simply be the standard of care. This is the vision. A world in which any child born with any genetic mutation in any liver-expressed gene can receive a custom-designed, precisely targeted therapy in weeks rather than never. A world in which the 7,000 rare genetic diseases that have no treatment are, one by one, addressed by a platform that can be adapted to each one. It will take time. There are formidable scientific challenges that remain, extending the approach to mutations in other tissues, not just the liver, ensuring the long-term safety of base editing in human patients, managing the cost of custom therapy development, which is currently enormous, building the regulatory frameworks that allow the necessary speed without compromising safety. But the door has been opened. KJ Muldoon opened it, and the people who walked him through it are already building what comes next. There is a specific weight to being first at something in medicine. It is not simply the weight of personal risk, though that is real and significant. It is the weight of uncertainty, the weight of not knowing, in a way that no amount of preclinical testing can fully resolve, what will happen when something unprecedented enters a human body. Kyle and Nicole Muldoon carried this weight. They were not scientists. They were not medical professionals. They were parents, facing an impossible diagnosis, an impossible set of choices, and the specific exhaustion of people who have been in fight-or-flight mode for months on end, trying to make the right decision for a child who cannot decide for himself. The consent process, the conversations between the Muldoons and the medical team about the risks and the possibilities of the treatment, was not a single conversation. Dr. Arns Nicholas has spoken about this specifically. Informed consent with parents making decisions about experimental treatment for a critically ill child is not a signature on a form. It was months of conversations, questions answered and then asked again, fears articulated and addressed. The same uncertainties returned too from different angles, because the stakes were too high for anything less. KJ Muldoon, in his public statements, described their decision with a simplicity that contains enormous depth. He said, "If you're going through something of this nature, it's important to find the tiniest bead of hope and hold on to that as long as you can." They found that bead of hope in the offer of a therapy that had never been tried. They held on to it through 10 months in the hospital. They were in the room when the infusion happened, sitting with the terror and the hope together while their baby slept. And after the announcement in May 2025, when the medical community and the global media understood what had happened, thousands of families reached out. Families of children with other rare genetic diseases. Parents who had been told there was nothing to be done. People who had been waiting, for years or decades, for science to catch up with the disease that was affecting their child or their sibling or themselves. Arons Niklas and Musunuru received, by their own account, literally thousands of requests. Most of them, they had to acknowledge, were not conditions they were currently in a position to address. But the fact of the requests says something important. The news of KJ's treatment had landed in the world of rare disease patients and families as something more than a medical milestone. It had landed as hope. Specific, newly credible, scientifically grounded hope. KJ himself is listed in Nature's 10 for 2025, a list of 10 people who most shaped science in the year. He is described as the trailblazing baby, the first person saved by personalized CRISPR gene editing. He is, as of early 2026, walking. He is meeting developmental milestones. His protein intake is well above what anyone thought possible for his age. His mother describes him as thriving. He does not know any of this yet. He is a toddler. He is interested in things that toddlers are interested in: movement, faces, sounds, the immediate sensory richness of a world he is only beginning to understand. One day, when he is older, he will learn that he was first, that his parents said yes to something terrifying, because a team of scientists offered them a bead of hope and they held on to it. That a team of researchers from Philadelphia and Berkeley and Cambridge and multiple companies worked nights and weekends and did in 6 months what they were told would take 18, because a baby needed them to. He will learn that he is not simply a patient who was treated. He is, in some real sense, the patient who changed medicine. In this series, we have told stories about survival in its most extreme forms. A soldier who fought a 30-year war that was already over. A woman who survived 3 sinking ships. A man who cut off his own arm and walked out of a canyon. A baby who fell from an airplane and walked out of the Amazon. These are stories about what human beings can endure, about the limits of the body and the will, about the strange, persistent, irreducible capacity of human beings to keep going when everything says they should stop. KJ Muldoon's story is different. It is not a story about endurance. KJ is a baby, and endurance is not a concept that applies to him in the way it applies to Aaron Ralston or Poon Lim. It is not a story about willpower or the refusal to surrender. It is a story about something else, something that connects it to a different thread in the human story, the thread of knowledge, and what happens when knowledge accumulates to the point where it can do something it could never do before. Every story in medicine has a before and an after. Before anesthesia, surgery was torture. After anesthesia, it was a tool. Before antibiotics, bacterial infection was a death sentence. After antibiotics, it was treatable. Before vaccines, smallpox killed hundreds of millions of people. After vaccines, smallpox was eradicated from the earth. KJ Muldoon's story is one of those afters. Before February 25, 2025, no human being had ever received a personalized gene editing therapy designed specifically for their unique genetic mutation. After February 25, 2025, one had. And the world that comes after that date is different from the world that came before it in the specific, practical, consequential way that all genuine medical breakthroughs produce. The children who will be born with CPS1 deficiency in 10 years or 20 will not face what KJ faced. They will face a different world, one in which a platform exists, in which the regulatory pathway is established, in which the manufacturing processes are refined, in which the knowledge of what is possible has been demonstrated by a baby in Philadelphia who wore an Eagles jersey home from the hospital after 307 days. And beyond CPS1 deficiency, beyond urea cycle disorders, the platform that was built for KJ points towards something larger. The 7,000 rare diseases. The children who are currently being told there is nothing to be done. The families who are holding on to whatever bead of hope they can find. The answer to those families is not yet, not yet. We cannot reach your specific disease yet. The platform is not extended to your tissue type yet. The cost is not yet manageable. The regulatory pathway is not yet routine. But the answer is no longer never. It was never before KJ. Now it is not yet. Not yet is a different universe from never. It is a universe worth living in, worth working toward, worth the nights and weekends and the 6 months of sprint and the 3-hour infusion while a baby slept and his parents sat in a hospital room with the weight of something unprecedented pressing on their chests. On August 1, 2024, a 4-pound baby was born in Philadelphia. His name was KJ. His liver could not process ammonia. His parents were handed an impossible situation and told to choose. They chose hope. On February 25, 2025, a therapy built specifically for KJ, designed from the specific letters of his specific genome, manufactured in 6 months by teams of scientists working with a purpose and a speed that is almost impossible to understand from the outside, entered his bloodstream for the first time. He slept. On June 2, 2025, he went home, down a hallway lined with the people who had cared for him for 307 days, through bubbles and familiar faces and the specific emotion of a medical team that had done something they would spend the rest of their careers understanding the significance of, into a police escort, into a car, into a house he had never been to. In his Eagles jersey. He is walking now. He is thriving. He is, by every measure available, doing what Bobbies are supposed to do, growing, developing, learning the world, demanding attention and returning it with the specific intense focus of a small person for whom everything is still new. He is not cured precisely. The disease still exists in his genome, and the therapy has not replaced every liver cell, and the long-term trajectory of his condition will require continued monitoring and probably continued treatment as he grows. But the severity of his disease has been fundamentally transformed. The ammonia crisis that was building in his body, that could have caused catastrophic neurological damage before a transplant was possible, has been interrupted. He has a future that, before February 25, 2025, was not guaranteed. Dr. Curran Musunuru, who was in the room when the first infusion happened, said of that moment, "I don't think I'm exaggerating when I say that this is the future of medicine." He was right. The future of medicine started with a 4-pound baby, and a team of scientists who worked nights and weekends, and a pair of parents who found the tiniest bead of hope and held on. KJ Muldoon is nature's trailblazing baby of 2025. He is also, and more simply, a child. A child who is home, who is walking, who is eating protein, who is growing, who is, right now, somewhere in Pennsylvania, completely unaware that he has already changed the world. He has time to learn that. He has time for everything. Thank you for watching. KJ Muldoon's story is the most recent in this series and the most forward-looking because unlike every other story we have told, this one is not finished. It is just beginning. If this story gave you hope, share it. Someone who needs to hear that not yet is different from never should hear it today. Subscribe for the next story, and leave a comment what part of KJ's story moved you the most: the science, the parents, the 6-month sprint, or simply the image of a baby going home in an Eagles jersey. I want to know.